So, good morning and welcome back. So, today we are here with the next part of the ABO discrepancies. And today we will be discussing about the confirmation of weaker subgroups in the group of A and B. So, often we encounter the weak subgroups of A and B in our day-to-day practice, and some subgroups of A have anti-A1 antibody. So I have Dr. Prachi Janoti, who is a first year resident at our institute, and she will be walking you through step by step as to how to identify these subgroups and how to address the transfusion concerns in these patients. So over to Dr. Prachi.
Welcome everyone. I'm Dr. Prachi Janotti and we'll be discussing the case of a 50 years old male admitted for laparoscopic cholecystectomy, no previous transfusion, surgical or significant drug history. We received his blood sample for pre-transfusion ABO grouping. The requirements of forward grouping are commercially prepared anti-sera, monoclonal anti-A, anti-B and anti-AB. Three test tubes labeled as anti-A, anti-B, and anti-AB. Beginning with forward grouping, we will add 1 drop of anti-A to the respective tube, 1 drop of anti-B to the tube labeled anti-B, and 1 drop of anti-AB to the respective test tube. This is the patient sample from which we prepared 3% red cell suspension. We will add 1 drop of 3% red cell suspension to all the 3 test tubes of forward grouping. Gently shake the tubes to mix the contents thoroughly. Coming to reverse grouping, we have 3 tubes labeled A cell, B cell and O cell. This is the patient's serum. We add 2 drops of patient serum to the 3 tubes labeled A cell, B cell and O cell respectively. These are the pooled A cells, pooled B cells and pooled O cells. We add 1 drop of pooled A cell to the tube labeled A cell, 1 drop of pooled B cell to the tube labeled B cell, and 1 drop of pooled O cell to the corresponding tube. Gently shake the tube to mix the contents thoroughly. Transfer the tubes of forward and reverse grouping to the centrifuge. Make sure to balance the tubes against each other. Centrifugation has to be done according to the manufacturer's guidelines. Coming to results of forward grouping, in anti-A1, there is a strong 4+ agglutination. In anti-B, there is no agglutination. In anti-AB, there is a strong 4+ reaction. Results of reverse grouping, in A cell, there is a 2+ reaction. In B cell, there is a 3+ reaction. In O cell, there is no agglutination. This is a side-by-side analysis of results obtained in forward grouping and reverse grouping. In forward grouping, in anti-A, there is a strong 4+ reaction. In anti-B, there is no reaction. In anti-AB, there is a 4+ reaction. The interpretation according to forward grouping is A group. In reverse grouping, in A cell, there is a 2+ reaction. In B cell, there is a 3+ reaction. And in O cell, there is no reaction. The interpretation according to reverse grouping is O group. Hence, this is a case of group discrepancy. In view of the group discrepancy, a fresh sample was asked from the patient and forward and reverse grouping was repeated at all 3 temperatures. The results of forward grouping showed the same result in all 3 temperatures and the interpretation being same as before, A group. The reverse grouping showed the same result in all 3 temperatures and the interpretation being as O group. Hence, this is a case of group discrepancy. In suspicion of a weak subgroup of A, we will proceed with further testing by adding anti-A1 lectin additionally in forward grouping and using A1 and A2 cells in reverse grouping. Beginning with forward grouping, we will add 1 drop of anti-A to tube labeled anti-A, 1 drop of anti-B to tube labeled anti-B, and 1 drop of anti-AB to tube labeled anti-AB. Then we will add 1 drop of patient red cell suspension to each of the tube. Additionally, we have taken 2 tubes labeled anti-A1 patient and anti-A1 control. To both the tubes, we will add 1 drop of anti-A1 lectin. In patient's tube, we add 1 drop of anti-A1 lectin and 3% red cell suspension of patient, whereas in control tube, we will add pooled A cells. Gently shake the tubes to mix the contents. Coming to reverse grouping, we will take 4 tubes labeled A1 cells, A2 cells, B cells, and O cells. This is patient serum. We will add 2 drops of patient serum to each tube. For reverse grouping, we have commercially available A1 cells, A2 cells, B cells, and O cells, and we will add 1 drop of each to the respective tubes. Gently shake the tubes of forward and reverse grouping to mix the contents thoroughly. Transfer all the tubes of forward and reverse grouping to the centrifuge, making sure to balance the tubes against each other. Then we will centrifuge according to the manufacturer's guidelines. Coming to results of forward grouping, in anti-A there is a strong 4 plus agglutination, in anti-B there is no agglutination, and in anti-AB there is a strong 4 plus reaction. In anti-A1 patient there is no agglutination, whereas in anti-A1 control there is a strong 4 plus reaction. Coming to the results of reverse grouping, in A1 cell there is a 2 plus reaction. In A2 cell there is no agglutination reaction. In B cell there is a 3 plus reaction, whereas in O cell there is no agglutination reaction. This is a side-by-side analysis of forward and reverse grouping. In forward grouping, in anti-A, there is a 4 plus reaction. In anti-AB, there is a 4 plus reaction. And in anti-A1 control, there is a 4 plus reaction. In reverse grouping, in A1 cell, there is a 2 plus reaction. In B cell, there is a 3 plus reaction. Whereas in A2 and O cells, there is no reaction. The conclusion is that this is a subgroup of A with anti-A1 antibody. We had another case of a 66-year-old male who was admitted for total hip replacement surgery. We received his blood sample for pre-transfusion ABO grouping. These are the results of forward and reverse grouping. In forward grouping, in anti-A, there was a 4 plus reaction. In anti-B, there was a strong 4 plus reaction. And in anti-AB, there was a 4 plus agglutination reaction. Hence, the interpretation being that it is an AB group. Whereas in reverse grouping, in A cell there was a 2 plus reaction, in B cell there was no reaction, and in O cell there was no reaction. Hence the interpretation being that it is a B group. Since there is a suspicion of a weak subgroup of AB, we proceed with adding anti-A1 lectin additionally in forward grouping and using A1 and A2 cells in reverse grouping. These are the results of forward grouping. In anti-A, there was a strong 4 plus reaction. In anti-B, there was a strong 4 plus agglutination. In anti-AB also, there was a strong 4 plus reaction. In anti-A1 lectin patient, there was no reaction, whereas in anti-A1 lectin control, there was a strong 4 plus reaction. These are the results of forward grouping. These are the results of reverse grouping. In A1 cell, there was a strong 4 plus reaction. In A2 cell, there was no agglutination reaction. In B cells, there was no agglutination reaction. In O cells, there was no agglutination reaction. These are the results of reverse grouping. This is a side-by-side analysis of results of forward and reverse grouping. In forward grouping, in anti-A, anti-B, and anti-AB, there is a strong 4 plus reaction. In anti-A1 control, there is a strong 4 plus reaction. In reverse grouping, there is a strong reaction in A1 cells and no agglutination in A2 cells, B cells, or O cells. Hence, the conclusion is that this is a subgroup of AB with anti-A1 antibody.
So thank you so much, Prachi, for taking us through the step-by-step procedure for identifying the weaker subgroups and identification of anti-A1 antibodies. So can you please briefly explain what exactly a subgroup is? Is there any difference in terms of quantity or quality of antigens on the RBC?
Thank you for the question, ma'am. The term subgroup refers to phenotypes with variations in the structure or number of the A and B antigens related to variations in the enzymes that produce them. A and B subgroups can result in either of the following: reduced number of structurally typical A and B antigens, or production of slightly altered A and B antigens which may or may not be reduced in number. Overall, A subgroups are much more common than B subgroups. Some individuals with an A subgroup may form antibodies to the typical A antigen, also known as the A1 antigen, because of either quantitative or qualitative differences.
So yes, very rightly mentioned that there is a structural and the quantity wise, and also there is a difference in the quality of the antigens on the red cell surface. So can you let me know what are the different subgroups of A and B which are encountered clinically?
The two principal subgroups of A are A1 and A2. Other subgroups are A intermediate, A3, AX, AM, AND, and AL. Whereas subgroups of B are B3, BX, BM, and BL.
Okay, so in the videos, as you showed, that you have used anti-A1 lectins. So can you please explain what lectins are and name a few lectins which are widely used in the day-to-day practice in the immunohematology laboratory?
Thank you for the question, ma'am. Lectins are carbohydrate binding proteins present in the seeds of many plants, fungi, bacteria, and in animals. Lectins bind specifically to the sugar residues which project from red cell membrane, causing RBCs to clump. This binding is as specific as antigenic recognition by antibodies. Many lectins are useful as blood typing reagents. Example: anti-A1 lectin derived from the seeds of plant Dolichos biflorus and anti-H lectin derived from the seeds of plant Ilex europaeus.
So as we saw in both the cases, there was presence of anti-A1 antibody. So if these patients require PRC transfusion, so what would be your advice? What group of PRCs should be transfused to them? And is a transfusion of group A PRC safe in these cases?
In case 1, where the patient is subgroup of A group with anti-A1 antibody, group OPRC should be used for transfusion, whereas in case 2, where the patient is subgroup of AB with anti-A1 antibody, group O or group BPRC should be used for transfusion. In both cases, transfusing A group will cause hemolytic transfusion reaction.
So thank you, Dr. Prachi, for taking us through the entire procedure of identification of weaker subgroups. And with that, we come to an end of this episode. Do subscribe for the next episode, wherein we will discuss about the secretor status. And if you have any questions or if you have any interesting cases you have come across and would like to share, please drop in to the comment section below. Thank you.